founder of Jan Biotherapeutics and a seasoned consultant, Dr. Brar has played a pivotal role in guiding companies through therapeutic development to clinical testing. Join her today in engaging on her presentation, New Treatments for Heart Disease, Viral Delivery and MicroRNAs. Let's hear it for Dr. Brar.
Thank you so much, Lee. Thank you, Professor Peterson, for that wonderful overview of treatments in women versus men for heart attacks. And you see there's a lot to be done. And also Dr. Indra Pulima's talk as well, which was really interesting. Is that better? Yeah, okay, right. So I'm going to switch gears a little bit. So we're moving from the clinic now back new treatments for heart disease. We're specifically going to be talking about viral delivery, where we're using viruses to deliver genetic material to correct a disease, and also an introduction to microRNAs. So statistics, why new treatments are needed. Every 34 seconds, someone in the United States is going to die of cardiovascular disease. That's 224 deaths per 100,000. In the United States, every year, there are 928,471 US deaths related to cardiovascular disease. Globally, that's 238 deaths per 100,000. Cost of managing heart failure in the United States is approximately $240 billion every year. It's the 12th most highest, it's 12% of the total health expenditure in the United States for all health-related diseases. And the problem with cardiovascular disease is there is no treatment, and the numbers are going to increase. We've heard in our previous talks, both the elderly population and also younger patients. Life's essential eights are not to smoke, physical activity, healthy diet, body weight, maintenance, sleep health now, control your cholesterol, control blood pressure, control blood sugar. There are also genetic causes of cardiovascular disease, and this is where viral therapy is coming into more of a play. What is viral delivery? The viral delivery is the use of safe and active viruses that can deliver DNA, RNA, and now microRNA drugs to treat specific heart diseases affecting select patients with genetic diseases mainly. So DNA is made in the nucleus. It controls is basically the genetic code of our human bodies. DNA is transcribed to messenger RNA. Messenger RNA is translated to protein. Protein is the functional units that work for our cells and also our tissues. MicroRNAs combine to messenger RNA. They were just found about 22 years ago. They're non-coding RNAs. They're about 20 nucleotides in length. They bind to messenger RNA. They prevent the messenger RNA from being translated into protein. So the human body has about 2,500 microRNAs, and they regulate about 80% of protein synthesis in our body. Huge regulators. The companies that are in gene therapy trials. So these are just some of the companies, and I'll go through each of them, that are developing new viral delivered treatments to treat specific heart diseases. They're recruiting patients now. They're using viruses, different types of viruses. Xylacor, they're developing a virus to express a DNA and then a messenger RNA and protein to encode protein called vascular endothelial growth factor that can promote blood vessel growth in a damaged heart. There's also treatments for Friedreich's ataxia that affects about 7,200 patients in the United States. So a rare disease that are overexpressing the DNA encodes for specific protein. Shannon's disease, this is a very rare disease. 350 US patients with that disease, they have hypertrophic cardiomyopathy, like phenotype. The Rocket Pharmaceuticals is developing that drug. And then there's also a number of other companies. Sarepta Therapeutics is working on a new viral delivery for the treatment of limb-girdle muscular dystrophy. That affects about 25,000 patients in the United States. Pfizer is in phase 3 clinical trials for a specific cardiomyopathy that affects about 20,000 patients in the United States. We have specific mutations. And then Lexeo and Rocket as well have other clinical trials ongoing. So the majority of the clinical trials are at different stages. When you develop a drug, firstly, you have to test it in an initial population, a small population, that's called phase 1. The FDA then requires you to test in a larger population where you can adapt your clinical trial design. That's called phase 1/2. There are also adaptive trial designs. Phase 2 is then required to increase your sample size and probably enrich your population with specific characteristics and also control the population that you enroll so your drug is effective in that population. And phase 3 is what we call a pivotal clinical trial. So when drugs get approved, they have to go through generally all of these clinical trials. Phase 3 is a larger population. Once the drug passes the phase 3 trial and meets the endpoints of that trial, the companies then can apply for a new drug application and then commercialize the drug. So you can see in the black circles, that's the number of patients in the clinical trials. The trials are not as big as they used to be now because of the significant unmet medical need. We need to get drugs to patients quickly. So there is a fast track process now where drugs do not have to have, especially for heart failure, tens of thousands of patients, where they use mortality as an endpoint. We're now using surrogate endpoints to see whether a drug is effective or not, rather than all-cause mortality, which can take 5 or 10 years to determine whether a drug is effective or not. There are also a number of companies that are targeting RNA, right? So all types of RNA. And these are just some of the companies on the screen here. Some of them are targeting messenger RNA to treat heart failure. Some are targeting non-coding RNAs, similar to microRNAs, and some are targeting other aspects of RNA biogenesis. Most of these companies are in the preclinical stage, except for Moderna. They're also working on VEGF. But let's target one particular case study. So Cardior, that's a company. They are developing, well, one of the first companies that's targeting a microRNA for the treatment of heart failure. They are targeting a microRNA called microRNA 132. It's being tested in patients post-heart attack. So 2 weeks after a heart attack, they're giving not a viral delivery, but synthetic oligonucleotide inhibitor to this microRNA in the blood. They can give it intravenously. And the mechanism of action does not promote cardiac muscle regeneration, but it targets other messenger RNAs. So the messenger RNA targets these 5 in particular. It showed limited efficacy in phase 1 clinical trials with only marginal improvement in heart function. What's our analysis? So there's a lot of players out there. So there's brand new biological therapies that are being developed using viral and RNA transfer into cells to treat specific cardiac diseases. There isn't currently a commercialized gene therapy to treat any cardiovascular disease, but it's just a matter of time. As you saw, there were companies that are in phase 3 clinical trials. Most of these companies are increasing the expression of a DNA to correct a protein in patients that have a specific genetic disease. None of these treatments regenerate heart muscle. A lot of the damage is already done, particularly in these genetic diseases that occur in some of the patients when they're very young. So even for Duchenne and other treatments, there are new treatments that they're being administered to younger patients, but there are also a population of older patients where they have heart failure. But do we think any of these treatments are going to solve the $240 billion problem that is just going to increase? Professor Peterson and Dr. Purnima also talked about ischemic heart disease. This is the largest cause of death of both men and women in the United States. Coronary artery disease is the #1 cause, and it accounts for 41.2% of all chronic heart disease deaths. Holy grail is regeneration of functional heart muscle. If we could regenerate functional heart muscle after a heart attack, we could then treat ischemic heart disease and prevent the majority of patients dying from heart failure. There is currently no treatment that can replace lost heart muscle cells after a heart attack. So ischemic heart diseases, we've heard, can occur by a heart attack where there's a blockage in a blood vessel, prevented blood and oxygen from going to the heart muscle. Heart muscle is permanently killed, is replaced by scar tissue, heart becomes weaker, and heart failure develops. Our progress is we are, as a company, targeting microRNAs. How can they be used to treat heart disease? MicroRNAs are made in the nucleus. They're exported to the cytoplasm. What they can do, once they're processed correctly, they can bind to messenger RNA and either promote messenger RNA degradation or prevent and cause translational repression to prevent proteins being made. So the technology that we've licensed is from the Salk Institute, and they found an innovative way to regenerate heart muscle that could potentially be a treatment for ischemic heart disease. It's by lowering microRNAs. We have to lower 4 microRNAs in combination. They're called 7A-C, microRNA 9900, evolutionarily conserved from zebrafish all the way to man. The solution is to lower these 4 microRNAs together. The mechanism of how they work is by lowering these 4 microRNAs, we're activating proliferation pathways to generate new heart muscle cells within the damaged heart and also promote cardiac myocyte differentiation pathways. This then generates new heart muscle cells, reducing fibrosis, decreasing heart function, and reducing mortality in patients. This is just some of the data. And so what the Salk scientists showed is that if you inhibit 4 microRNAs together, you can get spontaneous proliferation and beating of human cardiac myocytes in culture. So what we've done, we've combined both viral delivery and inhibition of these microRNAs to develop a new treatment. And we use a virus. And the virus is an AAV virus. It's called JBTMIR2. The virus is single viral. It prefers the heart. It allows for transient expression of our microRNAs. It's non-integrative and it's safe. And it's formulated for intravenous administration. We only need to give a very low dose. It prefers the heart. We conducted a study in pigs, female pigs with a heart attack. This was done at UCSD. We subjected pigs to a 60-minute heart attack where we use a balloon catheter to occlude the left vessel, the left ventricle, called the LAD. This supplies 80% of blood and oxygen to the left ventricle, 60 minutes. We deflated the balloon, and then we gave our virus or control virus after the heart attack, or and also 2 months after her heart attack, and followed the pigs for up to 3 months. Not working, but in our experiments, we showed that we could actually normalize heart function in the pigs with a heart attack. We also increased left ventricular mass. We had also normalized other organ functions. So if you're increasing heart function, you're actually going to improve the function of other organs such as the kidneys. We had no abnormal safety findings, and there's no abnormal pathology. And the good thing about this virus, and this is an experiment that we did in mice, is that you tag it with an enzyme called luciferase and you put it into mice, and then you can dissect the organs after. And from this experiment, we showed that the virus that we've selected prefers the heart and skeletal muscle as opposed to just going to the liver, where a lot of viruses target. It's very safe. We've tested the safety in over 300 mice and 5 pigs. We've had no abnormal safety issues. It's cardiotrophic. We've yet to conduct our GLP toxicology studies. So to summarize, we've developed a new treatment using viral delivery to inhibit 4 microRNAs to regenerate heart muscle within the damaged heart. This treatment will be used for ischemic heart disease that is caused by lack of functional heart muscle. Treatment will potentially reduce morbidity and mortality, and $100 billion used to manage the disease. Regenerating heart muscle is a holy grail of cardiovascular drug development. If we could do this, we could treat a number of cardiovascular diseases. Next steps is to file an IND and conduct our GLP toxicology studies, additional patents, and we have CDA signed with strategic partners, and we expect to dose our first patient by 2026. Thank you.
Thank you so much, Dr. Brar. I think we have time for one question. One question from the audience?
I just wanted to ask you, do you have any idea what RNAs these 4 microRNAs bind to?
Right, yeah. So the power of targeting microRNAs is you can modulate the expression of a number of microRNAs. These 4 microRNAs target approximately 1,070 messenger RNAs. This regeneration appears to be specific to the heart muscle. Pathways that have been identified is it increases the expression of a protein called Fint beta, which is involved in the RAS-RAF kinase pathway that's involved in cell proliferation. It also increases the expression of a protein called SMARCA5, which is involved in chromatin stabilization. So they're the 2 pathways that the Salk has identified. We've also done our sequencing on our tissues, and we found that downstream 2 months after treatment, getting an increase in messenger RNAs that encode proteins that are involved in heart muscle structure and function. So troponin T2, which is part of the myocyte traction machinery, increases in addition to FHL1 protein, which is involved in sarcomere reorganization. Muscle cell.
Thanks so much. So I think we have one more speaker before we break for lunch. And when we do break for lunch, Dr. Brar and Dr. Peterson will be around to take more questions during the break. Okay, thank you so much, Dr. Brar.